Hello, everyone. Welcome and thank you for being here today as we host our Pathways in Medical Genetics webinar. My name is Devin Simmons, and I'm thrilled to serve as the STEM Lyceums Program Manager. I'm excited to dive into today's event where you'll gain invaluable guidance for academic and career journeys, and I encourage you to take notes throughout the session to capture actionable tips that will assist you in future endeavors. More information on the Center for Excellence in Education. CEE was founded in 1983 by Joanne DeGennaro and the late Admiral H.G. Rickover, who is the father of nuclear navy and civilian uses of nuclear power, with a mission to nurture high school and university scholars to careers of excellence and leadership in STEM. CEE's mission is to nurture high school and university scholars to careers of excellence and leadership in science, technology, engineering, and mathematics, and encourages collaboration between and among scientific and technological leaders in the global community. You may visit CEE's website to learn more about the center's cost-free programs for students and teachers, such as the Research Science Institute, USA Bio-Olympiad, the Teacher Enrichment Program, and STEM Lyceums. More about the program CEE has to offer. First, we have the Research Science Institute, which is a highly competitive 6-week summer science and engineering program open to rising high school seniors. Juniors are eligible to apply as well. RSI is the first cost-free summer science and engineering program that combines on-campus coursework in scientific theory with off-campus work in science and technology research. Then we have the USA Bio-Olympiad, the premier biology competition for high school students in the United States. It provides the motivation, curricular resources, and skills and training to take them beyond their classroom experience to the level of international competitiveness. And since CEE began administering the USABO in 2003, every member of the USA Bio-Olympiad team has medaled at the IBO. And last but certainly not least, we have our Teacher Enrichment Program and STEM Lyceums. The Teacher Enrichment Program provides opportunities for middle and high school teachers to connect with leading experts from industry and academia, explore cutting-edge research and development, make meaningful professional links with direct benefits for you and your students. And then STEM Lyceums is the newest initiative at CEE, which hosts virtual clubs for high school students to engage in discussions and explorations of STEM concepts of career pathways, and provide scholarship and internship resources to students nationally and internationally. So please visit our website to learn more about those cost-free programs that CEE has to offer. And now, I will now introduce our speakers. Today, as our speakers, we have Dr. Rob O'Malley, whose PhD is a Public Engagement Associate in Personal Genetics Education and Dialogue, a program based in the Department of Genetics at Harvard Medical School. PGED seeks to raise awareness and inspire curiosity, dialogue, and reflection about genetics. He is an evolutionary anthropologist by training, with over 7 years' experience in inclusive public engagement with science and informal science learning. He is also a research associate at the Center for Advanced Study of Human Paleobiology at the George Washington University and has served as co-chair of the Education Committee of the American Association of Biological Anthropologists since 2022. Sorry. He earned a bachelor's degree in anthropology and zoology from Miami University, a master's degree in anthropology from the University of Alberta, and PhD in integrative and evolutionary biology from the University of Southern California. That is Dr. Rob O'Malley. And then we also have Dr. Mimi Shirahasuhiza, forgive me for my mispronunciation. She is an alum of CEE's Research Science Institute, which I just talked about, class of 1988. Is a tenured professor at Columbia University Irving Medical Center in the Department of Genetics and Development, where she is also the vice chair of education and the assistant dean of mentoring for the Vagelios Institute PhD Pathway Program. She earned a BA in molecular biophysics and biochemistry from Yale University. She earned her PhD from the University of California, San Francisco, and Harvard Medical School. She was a postdoctoral fellow in the lab of Dr. David Schneider at Stanford University. Her NIH-funded work focuses on using the fruit fly, Drosophila melanogaster, to understand a circadian regulated physiologies and behaviors that contribute to disease. She has published in many high-impact journals, including PLOS Biology, eLife, and Nature. She is also passionate about research mentorship and training, not only in her own lab, but also broadly in Columbia's graduate and undergraduate research program. I'm going to call her Mimi, and these are our two speakers. And now I'm going to let Dr. Rob O'Malley take over and share all of his wealth of knowledge.
Okay, thank you so much. Welcome to today's webinar. I'm really excited to be here on, oops, am I not? Oh, you can hear me. Really happy to be here on behalf of my program to talk about sharing and learning about genetics, engagement in the classroom and beyond. So our program, Personal Genetics Education and Dialogue, was recently rebranded. Our new name, we think, better reflects our mission, which is, as Rose said, was to inspire curiosity, dialogue, and reflection about genetics, empowering people with the knowledge and confidence to navigate an increasingly complex world. How do we do this in practice? By creating resources for formal and informal educators, hosting workshops, webinars, and other convenings, and seeking out opportunities for collective sharing and learning with diverse segments of society. And that can include educators, but also policymakers, members of the film and entertainment industry, faith communities, and more. And you can learn about our program at our website. And I'm hopeful that whether you're a student or an educator or someone who's just interested in this topic, that there's going to be things that I talk about in my time today that will be of interest to you. So I think it's important to acknowledge my positionality as I come into this conversation. You heard a little bit about sort of my academic background. I also want to say I'm a white, cisgender, and heterosexual man. I come from a middle-class background, and I am a US citizen. I speak English and some Spanish and Swahili. I am a parent, and I consider myself agnostic, though I was raised Catholic. And I don't have a formal genetics background. My academic background is on studying wild animals. But I do have a lot of experience over the last 8 years in facilitating constructive conversations about science across differences of perspective and worldview, including on many socially contested topics like human origins and evolution. So for our time together, I just want to outline a couple of very brief goals. First, to just review some forefront genetics and society issues. We'll have an opportunity to share about different perspectives on genetics and why genetics conversations are important. And I will warn you that this will be somewhat interactive, or I hope it will be. We'll consider some inclusive best practices and guiding principles. We'll apply some of these ideas rather, in a short exercise, and give you an opportunity to reflect on possible next steps. And we only have 25 minutes, so this is very much an introduction, and I hope you'll take things we talk about today onward. So some big takeaways right at the beginning that if you remember nothing else from this presentation, I hope you'll remember. First, the science of genetics is increasingly a part of our lives and in society, that everyone has something to contribute to conversations on genetics. And finally, many best practices in inclusive science engagement can support learning, reflection, and empowerment. I wanted to encourage us, and there'll be some discussion in Q&A later, some community norms that I think are really valuable in thinking about talking and sharing and learning about genetics include what I've put here. I encourage you to speak from your own experience, to own the impact of your words, to listen to understand, not to respond, to critique ideas, not people, recognize that it's okay to change your mind, and share the space. My presentation and some of the conversation we may have might touch on some sensitive topics, including applications of genetics, historical and ongoing, law enforcement and reproductive health. So I just wanted to give that acknowledgement. And also I want to note, I'm going to be having some interactive points. And if you have a window or your phone handy, you can go to that website at the bottom or use the QR code to scan polev.com /ROBOMALLEY 941. And hopefully you'll be able to access the poll, assuming it's working. I believe it's working. So I want to pause and just encourage you to think for a moment. I've used this term already a couple times, engagement. And maybe just think for a moment about what we might mean when we say engagement. Because I'm not saying education, and I'm not saying communication. I'm saying engagement. So just sit with that for a minute. What might that mean to you? In this context, when I say engagement, which is a deliberate choice, what I'm talking about in the context of public engagement with science is intentional, meaningful interactions that provide opportunities for mutual learning between scientists and members of the public. And mutual learning refers not only to the acquisition of knowledge, but also increased familiarity with the breadth of perspectives, frames, and worldview. And I prefer engagement over communication because communication sometimes implies one-way transmission of information or addressing perceived wrong or incorrect ideas in others. And I think engagement captures a more collective and open mindset and also signals intellectual and cultural humility and a willingness to share and learn, all of which I think is important for more inclusive conversations about genetics. So with that, I want to just pause, and I believe the link is working. If you could go to this poll at the top and just think about why is public engagement on genetics important, and just put in a short response there. I believe it is going. Yes, so you can respond. Just type if you go to that website and type in a response. Helps to learn more about past and future, sure, right? It's a way to think about where we could be headed, and also like where we've come from. That's great. Can people become aware of problems? Sure. So that might be medical problems. It might be, informing ideas about population histories, relationship between genes and disease. It can be valuable to maybe increase research participation. That's great because genetic codes are different. Yeah, these are great. So thank you for responding. So people see that genetics and its applications matter. And so genetics spans from very personal questions like reproductive health to impacts on the world. For example, genetically modified foods and other organisms. And so there's so many issues, and this is just a selection of recent headlines highlighting where genetics is intersecting with people's lives. Reproductive health, sickle cell disease treatments, control of genetic information, movement of genetically modified organisms into commercial sale so that home gardeners can access these technologies. Genetics as a field of science has always had a social, historical, and cultural context. So I think when talking about genetics, it's important to start there. Confidence and trust in science by the US public has been strong for several decades, which I think is a good thing that's worth celebrating. As of 2018, more than 9 in 10 Americans agree that science and technology will offer more opportunities for the next generation. More than 7 in 10 Americans see more benefits than harms from science. And worldwide, 7 in 10 people signal that the work of scientists benefits people like them. However, respondents in the US and Western Europe are much more likely to agree with the sentiment that sentiment, more than 8 in 10, compared to people in North Africa, South America, or South Africa, so that 6 in 10 or lower. And these results that I've just quoted date to 2018. Surveys since then have signaled that by some metrics, measures of trust in science, scientists, and medical professionals has declined. And in the US, attitudes have become more polarized along political divisions. Another bit of good news on genetics specifically, the US public's familiarity with basic genetics terms and knowledge of genetics concepts seems to have shown improvement over the last decade, according to a 2022 survey. However, attitudes about the pace of change and the applications of new genetics advances vary. People are generally enthusiastic and favorable about detecting, preventing, and treatment of disease and the way that genetics can help with that, but tend to be negative or skeptical about technologies related to human enhancement or selection of traits. There's often voiced concerns about privacy, discrimination, for example, in the context of insurance coverage or employment. And about half of US adults say that the pace of science and technology advances, including in genetics, is making life move too fast. It's also worth emphasizing that distrust of science and of scientists and of medical professionals is not an anti-science position. In many ways, it's a historically informed perspective. There's no shortage of examples of scientific ideas being leveraged against people, often marginalized and minoritized people, in ways that have done tremendous harm. For example, the Supreme Court case, Buck v. Bell, affirmed the right of the state, in this case, the US government, to forcibly sterilize people it deemed unsuitable for reproduction. And that decision took place in a period where eugenics, the idea that humanity can be improved by breeding better people, was a completely mainstream and widely held idea. And one that was embraced, in fact, by many major scientific societies and organizations around the world, including universities such as the one that I work at, Harvard, which were in many ways the heart of the eugenics movement. And there's a tendency, I think, even among the field of genetics, to view this history as only history, to treat it as kind of a regrettable chapter that we've all moved beyond. But I think this is incorrect. I think that science, including the field of genetics and related fields, can continue in some cases to be a source of great harm in practice and in application. Many injustices related to science and scientific research remain unacknowledged. Others are acknowledged but not fully addressed. And even well-intentioned research can and has been co-opted to support, for example, false ideas of racial essentialism, the idea that we can distinguish racial groupings in humanity very deeply and firmly in genetics. Unethical practices and activities in which often marginalized and minoritized communities have more often been subjects of research rather than beneficiaries of research still impact many individual and community perspectives on science and medicine. I live in Maryland, close to home. The story of Henrietta Lacks, whose cells were harvested without her consent and have been used for decades in medical research with no benefit, financial or otherwise, returning to her family, is one kind of infamous example. And as noted by Dr. Crystal Sosi, a Diné geneticist, and her colleagues, for Indigenous communities, harms from biomedical research are ongoing, are not just in the past, and take place within a research framework that's often about doing things to and about Indigenous people rather than for, with, or by them. So I hope I've just briefly shown you or made the case that genetics issues are ones that regularly touch people's lives, often in extremely personal ways, that genetics can intersect with values, identity, and worldview, and that this is therefore a field where there really need to be broad, inclusive societal conversations. And whether we're science educators, students, people who are maybe thinking about careers in the life sciences, whatever our role might be, we all have an important role to play inside and outside the classroom in supporting those types of conversations that really need to be happening. So what does PGED, how does PGED fit into this space? Well, whether we're fostering conversations at community science festivals, or at a mosque or a church, or in a classroom, or in a teacher workshop, some of the values we strive to uphold and model include being empowering, by which we mean a belief that people and communities deserve to be active participants in shaping the role of genetics in our lives and society. And we do that by promoting learning, discussion, and agency in decision-making about genetics. We strive to be people affirming, by which we mean affirming and respecting the humanity of every person. We try to make space for people to engage with different values, perspectives, and worldviews, and try to be guided by personal and cultural humility, vulnerability, and empathy. We strive to be accurate and authentic, ensuring that our work is grounded in science, but also recognizing that science is a process with a social, cultural, and political context. We also strive to model transparency, openness, and curiosity. And finally, we strive to be impactful. We want to support meaningful outcomes for people, communities, and society at large, and therefore focus our activities on things that are timely, relevant, and engaging. So again, even in areas such as health, where a link between genetics and individuals' bodies and lives can be very clear, we really always have to think about science or genetics in this case in the context of in a holistic way. We can't talk about genetics in health contexts without acknowledging all the other factors that might influence health and well-being, what are sometimes called like social determinants of health. And for many people, talking about aspirational health benefits of medical advances in genetics or otherwise can fall flat because precision medicine is not going to be of use to someone with no health insurance who can't even afford or have access to a doctor. And in the context of many transformative genetics advances, many people already feel at a distance, forgotten, or unheard. And we're really trying as a program to address those feelings of not being heard. So again, we're returning to some interactive activities now. So there's 3 questions, and it should come up now automatically if you still have that window open. And you can just respond, and there's no right or wrong answer. The idea is we're just trying to capture the range of views in our virtual space here. So this first question is, I would want to know my likelihood of developing a life-limiting genetic condition, so potentially life-threatening genetic condition, only if there are ways to prevent or treat it. Okay, so we have a range of disagree to agree, but nobody's strongly in either camp. There's some ambivalence. So for our next question, direct-to-consumer genetic testing. So those like ancestry kits that you can get from companies like, and I'm not endorsing these, but various companies that purport to tell you something about your ancestry and perhaps tell you something about potential health risks by examining your genetic code. These direct-to-consumer genetic testing can be a good way to learn about my ancestry and genetics-related health information. Often the answer can be it's complicated, but that's hard to capture in a five option poll. Okay, our final question. Law enforcement agencies should be able to access private DNA databases in their investigations. So maybe a crime or serious crimes have been committed. Law enforcement agencies are seeking to identify or perhaps exonerate suspects. They should be able to access private DNA databases in their investigations. And I should note that this is not a hypothetical, because this is happening now and was in fact a contributing factor in the Golden Gate, solving the Golden Gate serial killer case a few years ago. Okay, so we have some strongly disagrees, some people agree. Okay, so I encourage you, we can talk more about this in the Q&A, but just reflect for a moment. Were there any surprises for you to in terms of the range of responses? And maybe thinking about like people who responded differently from you did. Think about whether the range of responses is representative of maybe, the perspectives you might find in your classroom or in other kind of public settings, your faith community, your family. How did this capture or this align with other communities or spaces that you're in? So this is the kind of exercise we often do, and this can often be like a launching point for having conversations about some of these topics, doing further readings, sharing, learning, short films, things like that. This is the kind of activity we often do to sort of get people thinking and talking. But the takeaway, I think, is that conversations about genetics can be really engaged with really intimate parts of our belief systems and our relationships. And some of these conversations can be quite challenging. I mean, thinking about like law enforcement, for example, or thinking about ancestry. Genetics conversations can bring up heavy or even traumatic topics. Thinking about, for example, eugenics, reproductive rights, disability and ableism, gender and sexuality, the role of law enforcement in society, medical abuse, and many other, pretty intense topics. So with that in mind, I do want to just note in exploring these topics, sometimes things can get challenging or contentious, and this is, can be challenging in the classroom environment. Some advice on navigating that would be to consider your own positionality as individuals and as members of communities and institutions. Recognize that we all enter conversations with our own experiences and education that inform our perspectives. Strive to approach disagreement with curiosity over judgment. Think about, avoid making assumptions about what people believe and why, and give them a chance to explain. I also want to note that genetics conversations, particularly in kind of the polarized environment that we find ourselves in, can often lead to really hard, potentially harmful conversations. And I want to acknowledge that it's completely valid to prioritize one's own safety and those of others. In particular, no one should be put in the position to defend their humanity or right to exist. And if conversations about contentious topics start to move in that direction, it may be best to disengage and look for other ways to spend one's time and energy. And so I just want to acknowledge that can happen. Okay, so coming up on my time, so just revisiting some key takeaways. I hope that I've made the case that the science of genetics impacts everyone's lives, that everyone has something to contribute to conversations on genetics because genetics is increasingly intersecting with so many elements of our lives, our society, the world around us, and is shaping ideas about humanity and about our relationship with the natural world and how we want to engage with each other. And I think that best practices in inclusive science communication and engagement, which I've tried to both model and talk about here, can support learning, reflection, and empowerment, whether in a classroom setting or otherwise. PGED's work over the last 17 years, I'm just wrapping it up now. PGED's work over the last 17 years has included the development of a lot of lesson plans and resources. You can check all those out on our website. These are formal lesson plans, keys for teachers, webinars and videos. We're increasingly developing informal science engagement resources like booth kits and a tabletop game, which we'll be rolling out soon. I encourage you to check out our website. You can join our mailing list if you want to keep abreast of our activities. And if you can, spend a few minutes after the webinar to just think about if there's a way that you can apply what you've learned in your classroom or elsewhere. I also just want to note that our program is based at Harvard Medical School, but I live in Maryland, so I'm local to the DMV. And so if you're interested in, we do classroom visits, school visits, teacher workshops. So if that's possibly of interest, please feel free to get in touch. And this is where you can get in touch with our program and myself. And with that, I will thank you for your time and hand it back.
And I just want to say thank you, Dr. O'Malley, for your wealth of knowledge and everything that you mentioned. Especially, I really enjoyed how you did model an inclusive classroom and how these conversations can be had. So just want to thank you for that. And before I hand it off to Mimi, I just want to say, if you do have any questions, you can drop it in the question and answer chat. We will answer those questions after both speakers have spoken. And then so you can add as many questions as you want, but just be mindful that we won't get to them until the end of both presentations. So now with that being said, I'm going to pass it over to Mimi.
That was great, Rob. I actually learned a lot there. And I love the polls. That's such active learning. I love it. Since I'm speaking second, and there are probably at least a few kids out there, maybe get up, stretch, stand up, move around. I do this in the classroom, right? Just to get some blood flowing, and let's have some fun. All right. So I don't know how many of you had a hard time waking up this morning, but I did. This is a picture of my son, who is a teenager. Yeah, right. And he has a hard time waking up in the morning. Both of my sons actually have a hard time waking up in the morning, much more than when they were kids. When they were little kids, they would wake up at like ding, 5 o'clock in the morning. They were like super awake, right? This is a change. It's not something that is your decision, actually. The onset of puberty is correlated with a shift in circadian rhythms to an evening preference. So you will stay up later and you'll find it harder to wake up in the morning. That is biology and the subject of what we're talking about, or what I study, which is circadian rhythms. Now, circadian rhythms are sort of stereotyped oscillations in activity over a 24-hour period. So over the course of a day, right? If you put up a guy in a cave, and they have actually done this experiment, they put a guy in a cave and they said, record when you get up, when you eat, when you feel hungry, when you feel sleepy. And it turns out for about a month, even if he's got no light stimuli or no way to see what time of day it actually is, he followed about a 24-hour rhythm for about a month, right? And that's because you have an internal clock in your body. In fact, you have an internal clock in all of your cells that tells you what time of day it is, which is pretty fascinating. And it regulates pretty much everything about your body. So your sleep-wake cycles, that's a very stereotyped manifestation of your circadian rhythms. Your heart rate, your body temperature changes with the time of day, your liver metabolism, cell growth, gene expression, they're all circadian regulated, right? And so we're interested in this for biomedical research because loss of circadian rhythms is closely associated with many diseases, including aging, metabolic diseases, cardiovascular diseases, neurodegenerative diseases. In fact, one of the first signs of Parkinson's is actually loss of circadian regulation and sleep disorders. And that can happen even before you have motor symptoms. Right? So psychiatric and mood disorders are characterized by loss of circadian rhythms, cancer, and even infection. Right? So we know that circadian rhythms are associated with disease. Can we use this information to either prevent or ameliorate symptoms of disease? Right? I use Drosophila to study how loss of circadian rhythm and circadian regulated genes contribute to health and disease. Why Drosophila? So here I've got a picture of, I don't know if you can see my arrow. This is a fruit fly or Drosophila, and it's tiny because this is a dime. I took this picture. So here, if you can think of how big a dime is, like a fruit fly is really, really small compared to the dime. Okay. Why do we study all of this in Drosophila particularly? Well, because we can do a lot of experiments with Drosophila. They have a very short reproductive span and they have a very short lifespan relative to humans. So it's like, they live to be about 3 months old. So we can study aging in a fruit fly in a very quick time frame. With all of these things that I've been talking about, circadian biology, sleep, metabolism, aging, they're all really highly conserved, actually. And many, many people have worked out other disease models for human diseases in fruit flies. So we can do a lot of manipulations genetically of the fruit fly using the tools and mutants that are publicly available. And there's this about 100 years of research using fruit flies. So it's very exciting to be able to play with all of these tools. Okay. Right. So this is my lab. We study all kinds of different things. And I'm just going to say here that there are maybe 3 foci in the lab: metabolism, immunity, and sleep. And these are some of the people who are in the lab, right? When we study different aspects of physiology, most of the students will come in with something that they're interested in, right? Like aging, lipid metabolism. Jared was very interested in like how to lose weight. Just like, that's a thing that people are interested in. And he was like, I wonder if I can make the flies lose weight. And indeed he can, using a time-restricted feeding protocol, which is also called intermittent fasting. So we can model a lot of human behaviors, diets, and sort of disease models in the fruit fly. And that makes it a really powerful tool for understanding how individual genes might be contributing to that disease or behavior. All right, so our basic strategy is to manipulate circadian regulated genes to identify the physiologies that are involved in disease, whether they prevent disease or contribute to disease or ameliorate disease, okay? So from all this, I can tell you I'm a geneticist, but I'm not a medical geneticist. And I know that the title of this was about medical genetics. So I wanted to talk about the differences. Genetics is the study of genes and how they regulate behavior and physiology and disease states, right? So that's what I do. I use a model, what's called a model organism, Drosophila, to do this. Medical genetics is really, I got this from Wikipedia. I'm at the medical school, but I'm not a medical geneticist. So I thought I would look to Wikipedia to get the definition. Is the branch of medicine that involves the diagnosis and management of hereditary disorders. So I have friends at Columbia Medical School who are medical geneticists, right? Thought it was really interesting that included in medical genetics is human genetics, right? Which is the study and identification of human genes that are associated with traits and characteristics, including hereditary diseases. And so this is something that is hugely interesting right now because we have so much genetic information, right? We know all the genes that we have, or at least we know the DNA code for all of those genes, what's called the genome, right? We don't necessarily know what those things do, right? And they're slightly different between flies and humans. There's a huge amount of overlap, lots of conservation, but it is true that there are still many, many, many mysteries, especially with complicated behaviors and physiologies and disease states. And so untangling all of those things and seeing what's important in humans is a really important field. Finally, clinical genetics is the use of knowledge that's obtained from the human genetic research to diagnose and treat disease, okay? And these are all, like I said, incredibly important fields. So so medical genetics in particular is super important. And I just want to highlight, for example, Dr. Mary King, who was a human geneticist who discovered BRCA1, which is or BRCA1, which is a gene that underlies hereditary breast cancer. Until she discovered this gene and used genetics, looking at many families and people who had the disease, right? And looking at siblings, looking at parents, and looking at how this the disease traveled through the family tree, and then sequencing various portions of the genome and identifying a gene that was always present, an allele or a form of that gene that was always present in persons with disease and not present in persons without disease, right? You can look for that kind of strong association or correlation, and you can identify genes that are causal or are involved in the progression of that disease, right? And so she discovered BRCA1, and now we can use that information and actually screen people for the BRCA1 allele that is disease causing. And that allows people who might be susceptible to the disease to undergo treatment or undergo various diagnostic criteria at regular intervals so that they can catch the disease early if they end up getting it. Does that make sense? So this is a super, super important piece of work that has saved hundreds of lives, if not maybe thousands of lives, right? Again, very super important field. Now, I'm not a medical geneticist, so why am I a geneticist and not a medical genetics person? And using a model organism as opposed to studying humans? Well, I enjoy identifying genes that are associated with disease and also with behavior and other traits. I also really enjoy understanding exactly how they cause disease or change behavior or regulate traits or the inheritance of various characteristics, right? So in order to discover new things and kind of understand how things work, it's really fundamental and really useful to use hypothesis-driven experiments, right? And that means manipulating genes and seeing what happens in a lot of ways. That's what I do with the fruit flies. It's really hard to do with humans. It's not considered ethical. And so, the fruit fly is a really great model for a creature that is similar to us, for which the genome is also much, much simpler, and for which we can create experiments and sort of watch how they react, and sort of figure out the puzzle, right? So I'm a puzzle person. The reason I'm a geneticist as opposed to a medical genetics person is I really enjoy this part of the process of science, is like figuring out the answers to little questions. Okay. How did I get here? I was told that this was maybe would be of interest to people. My high school AP physics teacher, whose name was Doc. I mean, that wasn't his actual name, but we all called him Doc because he was a PhD, and he sent me to a summer research program. And when I say he sent me, I really mean he sent me. Like, I was so clueless, and this was many, many years ago too. And I was born in Hawaii, and I've only been off of the island like once, and I really had no idea that there was this whole kind of world of science out there. I was thinking of being a lit major. I was a real like English person. I wanted to be a writer. And honestly, back then it was one of those things where I think like girls were not encouraged to do science as much, and the sort of expectation was that I was more of a humanities person because I was a girl. And Doc basically said, "Oh, that's just not true. So I think you'd be a great scientist." And I was like, "Me? I don't think I'd be a scientist." Like that seemed very like, I don't know. That seemed like not me, right? Because I was this like young girl writer person. He sent me to RSI, actually, which is the sponsor of this whole series, and I loved it. It was the first time that I really engaged in science that was real experiments. It wasn't from a textbook, and it wasn't learning information, but actually going in and changing things and seeing what happened, right? And I I spent my summer as an intern at the NIH. This is when RSI was placed in DC and not in Boston. I found my tribe. It was a really amazing experience for me. Everybody in the program, by tribe, I mean the other kids who were there in the program. I felt for the first time like I fit in somewhere. And I'm still in touch with many of those people today, still, because they were so formative, actually. It was a formative experience for me. I changed my mind completely and decided I wasn't going to be an English major. And instead, I went to Yale and majored in molecular biophysics and biochemistry, which is on the opposite end. But it was really important for me to realize that science was an active creature in a lot of ways. It's like an organic process. It's not, it's not information that's set in stone. In fact, there's very little that we can say with absolute certainty about science, right? I mean, because there are things, little things that we add to it every day and things that we discover every year in every field, pretty much. So it's nice to know in a way that there's still so much ground to explore and that you can be a part of it. And that to me was really exciting. So for my PhD, I went to UCSF. And then for my postdoc, I went to Stanford. Now, how did I end up working on circadian rhythm? It's like a little bit of a strange topic in a lot of ways, right? I started working on circadian rhythms of infection because I'm a circadian mutant. I am a kind of an extreme night owl. So I have a really hard time waking up in the mornings. I think I just never grew up. I never like left adolescence or something. So what I did in my postdoc was infect flies with bacterial pathogens and then sort of looked at their pathogenesis. Something that I noticed, this was before I had kids, and so I was there in the lab very late at night, and I realized that the flies died really differently when I infected them at night versus when I infected them during the day. So 2 in the morning versus 2 p.m. was the maximal difference that I saw. I went to my boss and I said, this is really strange. Like, why would that be? I'm really controlling everything that I can. I don't understand why I'm getting this effect, and I'll show you the effect. It was pretty crazy. But on the left, on the y-axis, I'm showing you percent survival. So this is 100% alive, and this is 0% alive or 100% dead. And this is time in days, right, on the x-axis, right? And so the ones that are in gray are night flies, flies that I've injected at night, and flies in yellow that I've infected during the day. And you can see that this curve is like way above the other curve. So they're clearing the infection, and they're about 75% of them are surviving the same infection, and 25% or less are surviving that same exact infection. And the only difference between these two sets of flies is what time of day they think it is, right? So what I did to test this hypothesis that it was actually the time of day was I took myself, the experimentalist, out of the equation, right? He pointed out, my postdoc advisor, that I'm different at day and night. And so we built 2 boxes, that one of which had day, night, and the other of which went night, day, right? And so we took flies, separated them into 2 populations. One population went into one box and one population went into the other box. And these are siblings separated at birth. They're identical genetically, and they're on identical food. They're completely identical in terms of their conditions, except for one is thinking that it's night and day. The other one's thinking it's day and night, right? So they're in anti-phase with each other as far as their circadian rhythms. And I got this result. I pulled them out at the same time of day for me, the experimentalist, and injected them at different times of day for them, the experimental subjects, and found that actually there was a huge difference based on the time of day. And that told us that it was something circadian regulated. Okay, hopefully that makes sense. This was such a nice result that I ended up abandoning the other project that I was working on. And I've been working on differences between night and day physiology for like pretty much the rest of my career. We did find eventually the mechanism underlying this, which is that the blood cells in the flies, which eat the bacteria, were more active at night than they were during the day. And that's actually held true in both mice and humans, it's a process called phagocytosis by macrophages, and they tend to be more active during the rest phase than during the active phase of the animal. Okay, just to touch base again here with my lab, we do study immunity still and bacterial infection, but we've branched out into studying aging, neurodegenerative diseases. There are many models in the fly that are really interesting to us. We are really interested in sleep and the function of sleep because we don't know. We have some neuroatypical models, and we have genes that are associated with autism, for example, and that are conserved in the fly. We can look at the effect that that has on different behaviors and disease states. All right, so those are the kinds of things that we work on. I guess the question is, do I have any advice for young scientists? I would say I take this advice from Malcolm Gladwell, right? Why do I do what I do? There are 3 elements of job satisfaction that have to be considered, at least according to Malcolm Gladwell, right? Autonomy. So it's about how much autonomy you have, how much decision making, how much freedom do you have in your daily life, right? I have a lot of autonomy. Actually, too much autonomy can be sort of oppressive to a lot of people. So you have to just sort of find where your sweet spot is between structure and lack of structure, right? That's another way of putting it. You don't have a lot of choices now if you're in high school, and in college, you'll have a lot more choice, and you'll see how you spend your time is really important. And it's important to sort of think about what you like, what you want to do with your time, and how much freedom you want to have in terms of spending that time. Complexity is another thing. If you have a job that is too complex, then that's difficult. If you have a job that's not complex enough, that's actually pretty stressful too. So finding that sweet spot again of like where things are complex enough to keep you interested, but not overly complex to make you stressed out, that's a really important factor to think about in terms of your job, right? And then finally, it's compensation. And when people think about compensation, they often think about money, right? And that's an important factor. You do want to be financially independent, but it's also important to remember that compensation is the connection between effort and reward. You want to feel like you're getting compensated in a commensurate way between what you put in and what you're getting out, right? For me, a really big factor in that is who I get to work with. I get to work with students and other scientists, and I get to have like great conversations, and I get to mentor young scientists all the time. I find that really fulfilling. That's really satisfying to me. And so, for me, once I make a certain amount of money, I'm good. It's all these other things that make me happy in my job, right? And so these are just important factors to think about in terms of, if you're interested in science, there are many, many different fields within science that could make you happy. And you should think about pursuing the ones that satisfy these 3 elements of job satisfaction for you, right? There's no right answer. It's all about a match. What's good for you? Okay. And then I would probably say that my other big advice is to follow your puzzles, like whatever is interesting to you, right? Keep following your nose. I mean, that's oftentimes it's the things that affect people around us or affect ourselves, right? Like I'm interested in circadian biology because I'm such an extreme night owl. But there are many people that I know who are, for example, interested in cancer because somebody that they know and love had cancer. These are important to work on something that you feel is personally motivating for you. And then finally, just want to say, remember to thank your teachers. I go back to Hawaii and see Doc all the time. This is also my family. But my, he's one person who has inspired me like through the years. And he was the one who changed my life. So I go back and I thank him for that. Every time I go back. This is like once a year or so. And I think it's now that I'm a teacher, whenever I get an email, from a former student saying, this is what I'm doing now, and here's my update, I appreciate it, because I do love to hear that I've made a difference in somebody's life. Okay, and that's all for me.
Oh no, this was not me rushing you off.
Oh,
I just wanted to say thank you, Mimi, and especially that last part, having been a former teacher myself. For anybody who's in the audience, if you will make a teacher's day just by saying thank you. It doesn't have to be grand or anything like that, but a simple thank you or a kind gesture will you don't know the work that teachers go through.
yeah.
I'm sure Mimi, as you've learned, and I've learned myself, that it's a lot. So if you just give them a kind gesture, it'll make their day.
It
And
will.
so just want to say thank you again, Mimi, and thank you, Dr. O'Malley. I'm going to open the floor for any questions. I did have some questions myself for the both of you, and so hopefully that'll get the ball rolling if there's anybody in the chat. And if not, then that's okay. If there are any questions that don't get a chance to get asked, or maybe you're a little bit too shy, please feel free to email me and I can send those questions their way. They are very knowledgeable and they are willing to help. They took the time out today, so don't want you to miss that opportunity. And so my first question is for you, Dr. O'Malley. You were mentioning or law enforcement looking at private DNA databases and things of that nature. So my question is, on the ethical standpoint, is there any pushback that comes with that? Because I can imagine, as we saw in the poll, that there were a lot of people who were against that. So what is that pushback, and how is law enforcement sort of circumventing that so that they're not crossing any boundaries?
Yeah, so the context in which this comes up and comes up regularly, and I just want to emphasize, I'm not an expert, right? Like my role is often to kind of facilitate conversations. A lot of genetic information exists in, law enforcement databases, and that information moves into law enforcement databases. When people encounter criminal justice system, there's often very specific state or federal regulations in place that regulate how people's genetic information enters different databases that are controlled by law enforcement or that law enforcement has access to, how it gets used. Very often there are restrictions in place about how those gets used, how it's shared among agencies, things like that. What gets trickier is when law enforcement requests or has opportunities to access privately held genetic information databases that might be acquired by companies that are looking at ancestry or that are otherwise collecting genetic information, ostensibly for people who want to learn more about their genetics. But then that data is in a database somewhere, and sometimes there are company policies or terms of use in place that regulate how the company can share that information. Often there's some assumption that they're not going to sell your information or share your information. But what can happen is that law enforcement may come to these corporations and say, you know what, we're trying to solve, in this case, like the kind of the iconic example is the Golden Gate Killer, a serial killer, so killed a number of people over many years. And can we use this information to basically help us narrow down a possible suspect? And what's interesting is they didn't have the person, if I'm remembering the details correctly, the person in the database was not the serial killer, it was a relative of the serial killer. And so another dimension of this is that a relative who may choose to share their genetic information is also, to a degree, is sharing genetic information about everyone related to them. So the what we have to weigh in these situations, and I'll as another kind of element of positionality. So my wife is a forensic scientist. So my wife actually works with law enforcement to sort of understand, identify suspects from biological material, to in law enforcement context. So this is a personal thing that like my family, people close to me are dealing with. There is a lot of value in, solving crimes, serious crimes of violence, and
also identify victims.
And identify victims, right? There's a lot of value in applying these technologies to help families. And as I think many of us know and are aware, law enforcement and its relationship with many communities is fraught. There are real concerns about priorities of law enforcement, how law enforcement protects communities and their information and safety or not. And also concerns about, if I give my genetic information to a company, have I lost control of that information forever? So I don't know that there's like a right or wrong answer, but like what I would say is that these are complicated issues. And I think a challenge is, and like I have a PhD in biology, it is challenging to keep up with the state of affairs. It is challenging to kind of work through what it means to submit, it's interesting, right? I want to learn about my DNA. What's the harm in like sharing that? Well, actually, there are whole dimensions of this that I think it's hard to fully appreciate in the moment. And that I think that there's no clear answer, but it's important that we as individuals, as scientists, as educators, as people who are maybe thinking about moving into these fields, be thinking about the broader context of the work that we do and the kind of science we are a part of, which has potential for great help, but also some potential for real harm.
Absolutely. I couldn't agree more. I think that everything what you just said is true, and it is such a tricky subject to try to talk about. And, it may be that people aren't privy to what it's actually being used for, or just what they may hear. So it is tricky, but I think that having these conversations like we had today, and including what you said about having an inclusive space, I think that's important. And so Mimi, my question for you is, you mentioned the circadian rhythm, and you highlighted your son early on. And my dad has always said that there's a phrase where energy is wasted on the youth. And as we get older, we get more tired, especially as when we wake up. But when, like you said, as a toddler, some of them just pop right up like nothing has happened. So my question is, as we get older, why is that happening? Because I would love to pop up like a toddler at when I wake up for work. I raised my hand when I said it was hard for me to get up this morning. So I just want to know why is that happening? And I guess my question will probably be later, but if there is a way to sort of change that other than going to bed earlier, but what can we do for that?
Well, so a couple of things. There are a lot of cues that the clock will take its hint from. So, food and light are two major cues. So if you just like expose yourself to food and light at various times, then you can actually shift your clock pretty effectively. But the reason that we have changes in what we call chronotype, or if you're a morning person or a night person, and actually it switches back when you are elderly. A lot of times elderly people will wake up at like 4 a.m. and be like, oh, hey, I want breakfast now. And then it's like, 8 o'clock at night, and they're like, oh, I'm pretty tired, I'm going to go to sleep, right? So these are kind of stereotype changes. And then, of course, there's a whole spectrum of people in between, morning, afternoon, night people, or whatever. There's a really interesting hypothesis called the sentry hypothesis, where they gave actowatches, which sort of record activity of people walking around in these indigenous societies without sort of industrial light and so on. And they recorded them when they were awake or when they were asleep. And then what they found was that actually in the whole village, there was maybe an hour or two where nobody was awake, right? And you can see how that would be evolutionarily very useful, right? To have sentries, or basically people who are vigilant, are awake and alert, and like able to sense any kind of trouble, a fire, predators, disaster, all around the clock. So there might be a sort of a evolutionary reason why we've evolved. And most creatures, most animals don't have chronotypes that vary as widely as humans. So maybe there's a reason for that, and we should just accept and embrace the diversity of chronotypes.
Absolutely, just embrace everything. So that was all for my questions. It's looking like people are a little bit shy, which is perfectly fine. Want to honor the time that I've set out for this event, and just want to again just say thank you. If you do have any questions or anything like that, please feel free to email me those questions, and I can send them to both of our speakers, who are a wealth of knowledge in many different ways and many different fields. So just want to say that again. Thank you again to our speakers and our sponsors, including the Cloud Moore Charitable Foundation, for sponsoring this event. This was a wonderful event. And so if you want to continue to come to these events, please make sure that you sign up for and register for our event, so our sponsors can continue to get these shout outs. And then I just want to say thank you again for our audience member for joining us. And please visit the CEE.org programs page to register for the next webinars hosted by STEM Lyceums. Just register for our upcoming event, and we hope to see you there.